Welcome to episode number, I think, oh shoot, what are we at? I think it's 14. Yeah, 14. So look who's back. Look who's back with me. We got Josh Williamson.
Glad to be back, Adam. I'm not inundated with COVID testing at the moment today, so that's good.
That is very good. I didn't want this to become a trend, right? Where you don't join us. So this is great. It's good to have you back. This is how we, how it all started with the two of us. And then, again, is because we're on a small liberal arts campus with a lot of experts on various aspects of things. It's been great to have some guests come on. We're going to kind of go back to our roots though today, and just the two of us kind of talking about some of the science and with a focus on the vaccines today. So, so Josh, you're trained as a pediatrician, right?
That's right.
So vaccines are something that you have to deal with a lot. So why don't you open our conversation today, if you don't mind, with just sort of your kind of take on vaccines, as a pediatrician, and kind of things you hear about and deal with in the office.
How much time do we have, Adam?
Yeah, exactly.
Now, as a pediatrician, I can get a little bit carried away when it comes to talking about vaccines. A lot of experts think of vaccines as being medicine's greatest success throughout history, and I have to agree with it. I mean, it's amazing when you start to look at the details. I thought I did catch a little bit of the last episode. I didn't get to finish it yet, but the last episode was great, talking about flu, and there was a lot of discussion about vaccines in that episode, which I thought was great. I learned a couple things just in the first half. Some of that history kind of gets lost along the way, unfortunately. But vaccines are an amazing success, and with success, there's been some difficulty with vaccines, because as we are able to wipe things off the planet or really reduce how many people are affected by terrible diseases, you start to question whether or not you need the vaccine. And that becomes a very difficult part in all this, because as a pediatrician, a lot of times you have to explain to parents that, while you don't know anybody that was paralyzed from polio, we still need to give your child the polio vaccine so it doesn't come back. And that becomes a very difficult conversation sometimes. And kind of as you get into the weeds of the history of vaccines and the reasons for vaccines, there becomes a lot of questions. Ethical questions come up quite frequently. So as a pediatrician, you spend a lot of time educating parents on vaccines and talking about vaccines, and I think it's good. I think patients need to be an informed consumer when it comes to getting a vaccine. I think that's very important. I've met a lot of people that don't want to know details about vaccines. They just want to know what they should get, and that's fine. But I think for most of us, we should be informed. The bad part is that there's a lot of misinformation out there. There's a lot of skepticism, and there's and especially in today's society where there's a lot of opinion and it's sometimes difficult to tease out facts. It can be hard to be well informed. So a lot of people have to find an expert or experts that they trust and go with that information, because medicine is many times an art, and it's not always a science. So there's not always fine lines between the black and the white. There's a lot of gray areas, and I think we're going to get into that a little bit when we move forward in this episode.
Right. Yeah. Well, this comes up sometimes in my courses, like when I teach immunology. I kind of mentioned that vaccines have almost been too good in a sense, because we just don't, especially in westernized countries, we have, advances in public health, but also, sanitation and screens on our windows and good vaccines and antibiotics. And we just don't really deal with, family members dying at a very young age from an infectious disease anymore. And so we become very risk averse, right? Any amount of risk, no matter how small, we're just not comfortable with as a society anymore. And that's kind of an uphill battle right now the vaccine community is trying to deal with.
Yeah, I trained during an interesting time. I mean, there's always interesting times to train in medicine, but during my time in training, the Haemophilus influenza B vaccine had been introduced. So in pediatrics, meningitis in babies is a terrifying condition. So especially in training, you do a lot of septic workups, we call them, in babies less than, well, it used to be less than 3 months, then it was less than 2 months. Now there's a gray area between 4 and 6 weeks still. But anytime a young infant has a fever, you worry that the baby could have meningitis. You know, a baby can't tell you that she has a horrible headache and a stiff neck. So we have to worry about those conditions. And so spinal taps are the only way to know if a baby does or does not have meningitis, and that's part of being a pediatrician, especially working in the hospital setting. So in training, when you would learn that if you did a spinal tap and pus came out instead of clear cerebrospinal fluid, that was Haemophilus influenza B, until proven otherwise. That's what you would see when you do the procedure. So really, before you ever got the results, you knew what you were dealing with just by the clinical aspect. Mm-hmm. Well, the vaccine came out, and I never saw that. Even though I did hundreds of spinal taps, that was something I never saw. It was essentially wiped away. Now, that's not true. It wasn't wiped away. I mean, there are still cases, but they're they're extremely low. I think in the single digits across the country, as opposed to being a third of the cases. So that was an amazing success of vaccines, but that's not something that an ordinary person ever hears about. Haemophilus influenza B. Most people don't know anybody that lost a child to Haemophilus influenza B meningitis, or know anybody that was deaf from after they recovered from that condition. So so that was one success that happened right before I started. And then Rototech came in while I was in training. That's the vaccine against rotavirus, which we used to admit babies all the time for severe dehydration because of rotavirus. And nurses would tell you before you ever saw the patient that the diarrhea smelled like rotavirus. And nurses could tell you before you ever even got the test results back. And so fluid management was a huge part of what I did in my training and what I learned about. And then the rotavirus vaccine came out, and it didn't wipe it away completely, but the numbers of admissions for dehydration in babies dropped tremendously, and it's a huge success. And so, we have all these, I've kind of lived through these successes in that realm, but we had already wiped away the stuff that everybody else knew about. People had babies that died of whooping cough from pertussis, back in the 50s. These things have just kind of been picked off one by one. And now we're getting to the point where we might be able to develop a vaccine as fast as has been done here for COVID that was unthinkable. Really, even 5 years ago, I guess, definitely 10 years ago. So now with this new platform, as long as things work out the way it looks like they're going to, we could be entering a new era of vaccinology, and it's just very exciting.
It is, and it also speaks, Josh, to the benefit of like funding, whether private or federal, of just science, right? Basic science. Like all of this knowledge has been accrued and over the last 100 years. And so we can use that knowledge to really more efficiently hone in on a product. So, we're reaping the benefits of our tax dollars paying for this research, frankly. So yeah, obviously today we're focusing on the vaccines. There's been a lot of news recently about some results that have been released from several phase 3 vaccine studies. So we'll get to that in a moment. But Josh, I was hoping you could sort of give an overview of, well, what is phase 3? Like, what does phase 1 mean? What does phase 2? Just kind of give a brief overview of clinical trials, and then I'll go in and just talk a little bit about, for our listeners, about the different types of vaccine platforms that have been used so far.
Yeah, Adam, you do this so much better than I did. I know you've hit these in previous episodes with the phases of these studies, but you basically want to know whether a vaccine is safe or not, and you need to know how effective it is. So those are the processes, and as you move along each phase, you increase the number of patients that you have enrolled in the studies, because the events are sometimes very small. Now, it's difficult to tease out the numbers too, especially during a pandemic because you have to know how much disease is out there, and then you have to have so many cases in each arm of the study, the placebo arm and the treatment arm of the study, to see whether or not the vaccine is effective. Once you get to phase 3, you're looking at a larger population to see how effective the vaccine is, basically in real use, where the vaccine is going to actually be distributed. So it's basically like a smaller sample of how the vaccine is going to be used.
Right. So phase 1, I guess, is typically focused more on safety, right? You give it to a few people, a small scale study, just to see if there's any like obvious like negative events, right? And then
Right.
phase 2 is typically sort of like a dosing. For a vaccine trial, you might look at the immune response. You give this dose, what kind of antibody levels are generated. Give this dose, it changes. And then I guess phase 3 is essentially like how good is it? And you mentioned about placebo versus the treatment. Sort of the gold standard around the world is double blind placebo control, right? So can you explain that briefly?
Yeah, so that double blind, meaning that neither the patient nor the provider, the doctor, knows whether the patient got the vaccine or the placebo. I've seen from a lot of these studies the placebo is the meningitis vaccine.
Okay.
And so they do that because a lot of the vaccines have adjuncts in them to boost the immune response. So some of the vaccine trials, some of the anecdotal stories I've heard, some of the healthcare providers that have enrolled in the studies said that the next day they had fever, and soreness at the injection site, fever, and they were happy because they knew that they got the vaccine and not the placebo.
That's a good point. Yeah.
Yeah, but now they've tried to make the studies even better by having the placebo arm be the meningitis vaccine, because you would expect to get some of those side effects, which are very tolerable. 1 to 10% of people will generally get fever and soreness at the injection site, and maybe even a little local reaction. And so by adding a vaccine that we know the prevalence of side effects, and we know the safety of, because it's been out for over a decade, and millions and millions of doses already given, we already know what to expect with that vaccine, so we can use it as a placebo for the new vaccine.
That is really cool. Yeah, because you're right. I mean, probably 20 years ago or so, if you didn't have a reaction, you kind of knew you were in the placebo. So they're trying to control for that. That's really cool. So yeah. So and by the way, for people listening, if you feel like after you get a say flu shot, right? You go to CVS, get a flu shot, might be a little tender, maybe a little red. You might have a mild fever, maybe not. That in my mind is a good thing. That means your immune system is working, right? But if you don't get that, it doesn't necessarily mean, by the way, that your immune system didn't do anything. But it's just sort of a reassurance that, okay, I did get this shot. Yeah.
I can tell you a quick story there, Adam. That doesn't make parents feel better when their kid is screaming in the middle of the night because their arm hurts. Well, that means the vaccine's working.
Well, see, that's the
They
type of
don't
thing
like to hear that. I stopped saying that after I've been a
Yeah.
pediatrician for a couple of years.
Oh, that's so funny. Just purely in a scientific sense, that kind of stuff drives me batty. I mean, I'm a parent of young kids, so I, I can relate. That kind of stuff drives me batty. It's like, there's never going to be, or at least probably not going to be some sort of vaccine treatment that elicits zero symptoms. Like we have to kind of get over that. We're protecting our child from a potentially preventable fatal illness. Like, let's not get caught up on a few side effects and one night of a kid crying. I'm just, like I said, I can relate. I have 2 young kids, but let's not focus on that too much. That's just my opinion.
I'll tell you, I was blown away just this morning because I really like Medscape. I like that website, but they put an article out. There's a nurse wrote an article in one of the trials and was talking about the high fever that she had and the achiness, soreness at the injection site. And she did it. She had a very high fever, 104,
Okay.
Very high fever.
Which is, by the way, rare. I mean, that is not the norm. That's a rare side effect.
Right. But also not one to be alarmed about. I mean, it's not pleasant, but it's not one to be alarmed about. But in the middle of the night at 104, as a healthcare provider, she was very nervous and scared, and as we all would be. But but now this article makes it gets published today, and it's kind of like, okay, yeah, we need to tell people to anticipate the side effects, like you said, a low-grade fever, soreness in the injection site, and let's not get too worked up about that.
And that's probably on the order of 95 out of 100, injections are going to be very, very, very mild, right?
Right.
And you might have 1 out of 100 that you have a fever that high, or even maybe not even that high. But yeah, you pop a couple Tylenol, you put a cold compress on, like but. So this is kind of a sidetrack, but there are certain people that might have a negative reaction that we just don't know about because they have some sort of genetic, rare genetic abnormality, right? And this is an interesting thing to think of, futuristic now. I almost wonder if we fast forward even potentially a few decades, if everyone will just have their genome sequenced at birth. So we'll know, oh yeah, little Johnny here has a mutation at codon number 3 of this immune protein, so that's contraindicated for getting this vaccine, right? So I'm almost wondering if we'll have more information down the road on those types of things. But you can't not do vaccine campaigns if there's literally a 1 in a million chance that a kid might have a more severe reaction, right? But unfortunately, there's only so much we can know ahead of time, whether or not you're going to be potentially allergic or have some sort of really, really rare reaction because of some genetic predisposition, And
that is what I tell parents. There's a 1 in a million chance of something really bad happening, and that is a risk. That we were talking before the show about Paul Offit. He talks about, it's not no risk, it's less risk. Dramatically less risk with the vaccine, but there is it is the risk that we take.
But the risk of not getting the vaccine is
much greater. Absolutely.
So
yeah, and then there was an article, and I have not been able to locate it since, but there were some researchers that wrote a paper, and this is one of those things you read you see an article and you wait to see the after effects later, and I never saw the after effects later. So whether or not that publication, the larger studies didn't bear out, which is probably what happened, but there were some researchers that claimed to identify a genetic marker for people that had higher risk for side effects from the flu vaccine. And just like you said, Adam, I thought that was going to be the greatest thing. I thought that that was I thought it was happening then.
Yeah.
And this was a few years ago, but it didn't seem to pan out just yet. But I do think that you're right. I think that that'll be in the future, that we'll be able to better inform patients of their risk when it comes to these things. And that is the hardest part. I shouldn't say the hardest part. It's another difficult decision to make when you think that a patient may be higher risk for a side effect from a vaccine. And that's where the herd immunity comes into play. Where if as a society we all buy into it, then the vaccines are even more effective, because even if a vaccine is 90% effective, that's tremendous, and that's actually what we're seeing. But with the COVID vaccines, but that means 1 out of 10 people that takes the risk to get the shot isn't going to get the benefit of immunity. But if you have enough people doing it, then the disease isn't going to be around to catch, and you're going to protect those people that did take the risk, but didn't get the personal benefit. But as a society, we all get the benefit.
That's a good point, right? Because the vaccine, right, might not work in every single person. That's true even with like measles vaccine, potentially COVID, like you mentioned. There's also kids that, you know, we know they can't get it. Maybe they have had a pediatric cancer, certain, you know, individuals in organ transplant, they're on immunosuppressive therapy. There's certain vaccines they cannot get. Like, for example, the chickenpox vaccine, right? I'll get to this in a minute. That's a replicating virus. It's weakened, but it's replicating. And certain individuals, if they don't have an immune system, that can now become a pathogenic disease-causing infection. Whereas in, you know, someone with a fully intact immune system, it just gets fought off immediately, right? So there are certain people that we do identify, we do know can't get certain vaccines. And so you're right, the herd immunity thing is really a public service, right? To protect people that cannot literally get vaccines. So I also wanted to mention that you use the word adjuvant. So that's something that we should just touch on briefly. So I do mention this in my class as well. It comes from the Latin for, I think it means to help, adjuvare. And it basically is sort of like an ingredient or an additive that's in the vaccine to basically essentially boost the immune response to the vaccine. So without that adjuvant, you're not really going to get much of a response. It's not going to be very effective at all. So, you know, so like for the flu shot, for example, we'll probably keep coming back to the flu shot. You get a little bit of swelling, maybe a little bit of redness. That's all inflammation. That's pretty much caused by the adjuvant. The actual, you know, flu proteins in there aren't really doing that. It's that adjuvant that's boosting that immune response so that you get a more robust response to the vaccine. So essentially, for the meningitis vaccine you mentioned, as the placebo, not getting the coronavirus vaccine, but getting something else, that adjuvant in there is causing that immune response so that the person has some sort of symptoms that you're mentioning. So I was also reading, just very brief, I was also reading that some people are getting antibody tests done so that if they get the placebo or the vaccine is going to elicit antibodies in the person. And so some people are getting antibody tests to see, do I have antibodies to coronavirus? And if they do, they know where they can deduce that they were in the vaccine arm and not in the placebo arm. Either that or they were infected and didn't know it with actual coronavirus. Either way. But did you want to add anything else, Josh, on that before we get into vaccines a little bit more?
You just made me think of one other thing. It's interesting. All these volunteers that volunteered for it. You know, now we're trying to the CDC and the CDC is looking to, is putting forth the recommendations, and then each state's health department is trying to decide who's going to get the vaccines first. And some people are saying, hey, let's not forget the people that are in these studies and got placebo because
right.
they really earned getting the vaccine. They wanted to get the vaccine in the first place, and we shouldn't forget about them, and we should allow them to get it.
And they were helping society. They were doing something good for us by doing that study. You're right. That's a good point. Yeah. So what I'm going to do is just kind of give everyone an overview. I'm going to try to make this brief, of different vaccine platforms. There's actually quite a few. So historically, and we're going back, to the first vaccine. So like Max Tyler in I think the 30s developed the yellow fever vaccine, won the Nobel Prize for it. So that vaccine was passaged through multiple cells, multiple organisms, and it became weakened. So in the scientific community, medical community, we call that attenuated. It just means weakened. So that is one example, or one way historically that we made a vaccine. Take the actual pathogen and just weaken it. So you inject it into somebody, and the disease-causing nature of it has been reduced, right? And like I said, historically, one of the ways to do that was to passage it through cells. So for example, like you could take like smallpox virus, passage it in a petri dish, so it's outside of the body, just you passage it, let's say 500 times. At the end of that 500 times, it's going to be a very different virus than what it started with. And in theory, it's going to lose some of those disease-causing capabilities because it's divided so much in the absence of the actual organism. And that's the idea. It's become somehow weakened. Today, now with modern science and with technology, we can do it a little more targeted. Like we can remove genes that we know cause the disease and just remove those immediately instead of having to do this whole passaging thing. So that's typically what's done today. And the other historical way to make a vaccine is to inactivate it, or sometimes people just say a killed version. So inactivated or killed. And you might treat it with UV radiation to basically just zap the genome, and it mutates the crap out of it. It can't replicate anymore. You could heat kill it, or you could treat it with a chemical like formaldehyde. And of course you rinse that all away. You're not actually injecting excess formaldehyde into somebody's body anymore. But so either chemical treatment, heat, or some kind of radiation to inactivate it, so that you're getting the actual pathogen, just like with the attenuated. It's just either a weakened form or a completely replication defective form, so it's killed. So those are most of our vaccines today, are either attenuated or killed. And in the last probably 50 years, starting maybe 50 years ago, they're trying to come up with different things. So one of the ones now is called subunit. And what I tell my students is sub means below or part of. So it's not the whole thing, it's just part of it. And so you're injecting usually like a protein or a couple of proteins from the pathogen. So not the pathogen itself anymore, not the organism or the virus, but a part of it, a subunit. I should also mention, before we go any further, that the most popular vaccine, what's called an antigen, that's the thing that's actually going to induce the immune response in the person. So the immune response is going to attack a part of the virus, in this case coronavirus, and the antigen or the protein from the virus that's been the most popular choice is called the spike protein. So if you imagine the virus is sort of like a sphere, you have these spike proteins. I'll use my pen here, right? So here's the virus, and you can imagine these spike proteins sticking out from the outside of the virus, sticking out to the exterior, and those are the spike proteins. And that's what for example, like an antibody that's floating around in your body, in your tissues, your bloodstream, it can latch onto that on the outside of the virus and prevent it from getting into a cell. Right. So subunit is the third one. Another one that's been in development for several decades is called a recombinant vector. So the word recombinant, right? So the word combine is in there. And so what I tell my students is it's sort of like taking parts of usually two different things. So two different things, combining them together. So for, a common example that we'll come back to in a moment is taking a virus called adenovirus, and you basically strip away some of the adenovirus proteins from the or you take away some of the genes that make those proteins. You strip away from adenovirus, which typically causes, just basic common cold-like symptoms. It's a pretty innocent virus for the vast majority of people. That's why it's chosen. So you take this innocent virus, you strip away some of its genes, and then you put into it some genes from something that is harmful. People have tried that for HIV, for Ebola virus, and now for coronavirus. So that's called a recombinant vector. And then the other one on my list here that's of note for viruses is called nucleic acid based vaccines. Nucleic acid, so I'll just run through that briefly. So people, usually know that DNA is our genome, right? Our, genetic code, if you will, is stored in our DNA. It makes all of, makes us who we are, makes all of the proteins of the cell. And so the DNA is copied into mRNA. So you start with a gene, DNA, in the nucleus of a cell that's copied into what's called messenger RNA. That then leaves the nucleus, goes out into the cytoplasm of the cell, where it's translated within the cytoplasm to make proteins. So for example, for coronavirus, it gets into the cell, it has this, spike protein gene. That gene is translated in the cell to the spike protein, right? So it starts as nucleic acid and goes to the protein, right? Now, one thing that I should mention. Let me try to show you something real quick here. Did you want to jump in a second at all, Josh, to maybe add anything or ask any questions?
No, I wanted to mention that sometimes they also use viruses that cause disease in animals instead of mild disease in humans as well, right, Adam?
Yeah, so that might be an example of like a recombinant vector, where you take, like say a cow virus or something, and plug in some genes of another virus and use that as a delivery vector, or you're right, it can be used as a straight up attenuated virus. So for example, the smallpox vaccine, right? Like it originated, people think either with horsepox or cowpox, but there's enough like cross recognition, cross reactivity, so that you're vaccinated with cowpox and you raise an immune response against cowpox, but there's enough recognition or cross reactivity to recognize the human smallpox. Right, so you're right. So the animal virus is not going to cause the disease, so that's an example of sort of an attenuated version. Yes, that's a good point. So what I wanted to do is just show just a brief diagram, something that a student might see in in a course or something, and this is something that might be taught in like sort of intro biology. But the idea is that in eukaryotes, so like so here's a decent image here. Let me share my screen. So this is just from a website that just did a Google search from here's an image from Pearson, but you can see that in our cells that we have a nucleus, right? Here's our DNA, which has our genes. It's transcribed to make the messenger RNA that then goes out into the cytoplasm, translated by what's called a ribosome to make the protein, and that's what gives the cell shape and structure and enzymes that that do work for our cells and allow us to do all kinds of things. So again, in our cells, it starts as DNA, goes through this mRNA intermediate or RNA intermediate, and ends up as a protein. Just to remind everyone, coronavirus is a bit odd in the world of biological entities. It has an RNA genome, not a DNA one, right? So most of the world is plants and animals and bacteria. All of that has a DNA genome, whereas viruses are the only known entities out there that can have RNA as a genome, right? So flu has an RNA genome, polio virus you mentioned earlier, Josh, RNA genome, and coronaviruses have an RNA genome as well. So there's no DNA involved at all in the replication cycle of a coronavirus. It's RNA, and then it's basically some of that's going to be translated to make protein. So it basically starts at RNA and then goes to protein, where it can make new virus particles or make that spike protein. And so we just jump right in here. So one of the vaccines that is currently in development, and what we've been hearing about, so this is a great website from the New York Times. And so one of the things, so from Mrna and Pfizer, their platform is to use mRNA that's going to give rise to the spike protein of coronavirus. It's housed in this sort of like lipid, sort of like a, like a greasy particle. And that's just basically a delivery tool, because what's known, Josh, is that mRNA is by itself, unless it's somehow protected, is very unstable. It's gonna have a relatively short half-life. And so this sort of particle is used as a delivery way to get it into a cell. And so the shell of this thing, of this particle, delivers the mRNA directly into the cytoplasm. So again, you're not really involving the nucleus at all. So the nucleus, again, is where our genes are, right? Where the DNA is. You're essentially bypassing that transcription step. You're not giving the cell a gene or any DNA. You're giving it this sort of intermediate. So this mRNA, this messenger RNA, is immediately in the cell, in the cytoplasm, and then it gets translated right away, and to make the spike protein. And by having the spike protein now in the cell or in the body, the immune system can recognize that and start to mount an immune response. And I should also mention that the particle itself, from what I understand, can act as an adjuvant itself. So you're sort of getting that immune boosting thing that you need from this adjuvant with this particle. And so again, the Moderna and the Pfizer vaccines, you are using what's called an mRNA nucleic acid based platform. Okay, so one of the advantages in my mind is that this could potentially be a relatively safe vaccine, because you're not injecting, you're not injecting any kind of actual pathogen, virus particle, anything like that. You're just injecting this mRNA intermediate. So you're going to get this sort of like this burst of protein production, but then relatively quickly thereafter, I'm gonna just throw it. I don't know the answer for sure. I'm gonna throw out hours, let's say, hours to maybe a day of production, and then this mRNA is going to have a natural like half-life, a natural turnover. This mRNA from the vaccine will eventually be degraded within a relatively short period of time. So you get your burst of spike protein production from coronavirus, and then the mRNA that's causing that to be made is going to be degraded eventually. So that's an overview of these mRNA-based vaccines. So Josh, do you have any questions? Do you want to add anything?
I mean, that is a question I have, how long does that mRNA sit in there, and how long does it produce those proteins? And then, Adam, do those proteins, they only get presented onto the cell, we think? There's really no way for them to just release unless the cell dies, I guess.
There's going to be some cell death eventually. Yeah, so you will have some of those things floating around, but from an immunologist perspective, that's a good thing. The more cells to take up those proteins, the better.
Which might be why these vaccines seem to be so effective. I mean, from the early results, they got, they were hoping for greater than 50%, and they're reporting closer to 90% efficacy. So, so that is definitely a good thing.
And they've also reported that the levels of antibodies generated from the vaccines are either at or surpass the levels seen from like a natural coronavirus infection. So, which is another good thing.
So basically I don't think we know this, but I would like to hear your take on it. Every cell that starts to produce those proteins will be recognized by the immune system, then produce antibodies, but then also be attacked by the immune system, and that's part of it, right?
Okay, you're right. Yeah, it's a good question, right? So don't forget, so like somebody that's never seen this before, which is the vast majority of people, right, that are going to get this vaccine, you're not going to have like pre-existing antibodies to the vaccine antigen, in this case, the spike protein, right? The whole idea of this is to induce those, right? So, so you're going to get this initial burst of spike protein that will then eventually lead to the induction of antibodies and also T cells. And we had a previous episode where we talked about antibodies and T cells. So, so I think that was episode 3 or 4. So people can reference that. I don't want to end up being here for like 2 hours, right? But I could talk about this stuff forever. Right. So if you got like a booster shot, in theory, that second booster shot might be a little bit less effective potentially, because as soon as that gets into the cell, starts to make that spike protein, it's going to be recognized and potentially removed by the immune response generated from the first shot, right? And we call that immune memory. So,
Adam, do you think it matters what cell takes up the vaccine in these cases? I think that was an interesting topic I heard.
Well, there does seem to be evidence in their animal models that, so there's this really important cell called a dendritic cell. It's sort of like I don't know, some people call it sort of like a mastermind, or like the general, like to use a military reference, right? Sort of like the general of the immune response, it sort of initiates and leads the production. But yeah, so this DC or dendritic cell is more than likely taking up some of these particles or ingesting some of the debris from cells that have died that are making these antigens, and they're probably kickstarting this robust immune response. So I think they know that from some of their early work, some of these companies, and there's a lot of data that the companies don't release, but a lot of this work has been done in mice or monkeys, and probably some in humans as well, looking at more like the mechanistic stuff on the immune side of things. But for right now, I'm most interested in what are the levels of antibodies that are generated from this in a human, right? And what are the safety? But again, just, so to go back, right? So one of the historical ways to make a vaccine was to just weaken it, right? So it's still, the pathogen is still in your body. It's still a pathogen. It can replicate, it's just weakened, right? But in some people, if you don't know they're immune compromised, it could be a problem, right? Because now a weakened virus all of a sudden isn't so weak anymore when it's not faced with a good immune response, So in my opinion, just looking solely at the science, I think the mRNA vaccines could be extremely safe, because you're not injecting an actual virus particle, you're not injecting a bacterial cell. It's simply just the nucleic acid. It's basically just giving the code to make the protein. It's going to be degraded eventually. It's sort of self-limiting, right? It's not replicating. It's not making virus. It's not making any sort of replication entity. It's just making some protein for a short period of time, and then it's going to go away. And so again, just looking from what I know and just looking at my, what I've seen, I would say this would be potentially extremely safe versus something like a weakened form of a virus. But one of the issues is we don't know, right? You were mentioning this earlier, Josh, that there is no licensed vaccine using this technology. So there is an unknown for sure. But I mean, I would get this vaccine. I would have no problem getting this vaccine. I should also mention that, these companies in the federal government, et cetera, they're constantly monitoring for adverse events, even well after things are licensed. So it's not as if the safety monitoring goes away. It's always happening. It's always happening. But yeah, I can just highlight here briefly that, the Mrna vaccine, again, and this mRNA vaccine, just like the Pfizer one. So it was published in the news, 94% effective, right? So how did they get those numbers? So just to kind of give the listeners an idea here, so they were monitoring their clinical trial, which had tens of thousands of people. And Josh mentioned some placebo, some had the actual vaccine. And there were 196 cases as I think it was November 30th, I'm getting this data from. 196 cases, but 185 of those were in the placebo arm of the study, and only 11 in the vaccine group. And so if you just do the math there, it's approximately 94% effective. And interestingly, and this is important, that in the 11 that got the vaccine but still got coronavirus positive test result, there were no severe cases of COVID-19 in those people, whereas there were some serious events that happened in the placebo arm. So this
that's an important point, Adam, because that's often the goal. Often the goal of a vaccine is not necessarily to prevent infection, at least not entirely, but to prevent the severe cases and the bad outcomes that come with these diseases.
Exactly, and I think that might have come up with Hillary last time. That was a great episode. I really enjoyed that conversation with Hillary. But when they release these numbers, right? The flu vaccine this year is 40% effective. What they mean is that it's 40% at completely preventing infection, as Josh has mentioned, right? When people call that a sterilizing immunity. So the virus goes in your body, but it doesn't go anywhere. But you still get the flu vaccine because it's going to prevent severe disease in most people, right? So 40% effective means that it only completely prevented infection in 40% of people. But what I wish they would publish is somehow a percentage of people that did not get severe disease, because I think that's also important to know, right? So protecting events against infection altogether, or preventing against or protection from disease is also really important as well. So I'm glad you brought that up. All right, so that's mRNA, right? So those are the two kind of ones in the lead right now are the Moderna and the Pfizer. Both are these mRNA vaccines that make the spike protein. According to this site, again, from the New York Times, it looks like 4 that are recombinant vectors that are in phase 3. And all of them are using versions of what's called adenovirus. So there's a Russian company, there's a Chinese company, I believe it is. There's Johnson & Johnson and AstraZeneca in collaboration with Oxford. They're using adenovirus. So 3 of them are using human adenovirus, and the 4th one, AstraZeneca, Oxford, they're using something called ChAdOx. So ChAd. So the CH stands for chimp adenovirus. And the reason they're using a chimp adenovirus is that there should be no pre-existing immunity in humans to that virus. So again, there's nothing to kind of limit the replication or the effectiveness of the vaccine virus, in this case, the adenovirus, because there is no immunity in the human population to chimp adenovirus. So that's the reason they chose that instead of a human adenovirus. Sometimes pre-existing antibodies to the vaccine virus. Remember, the adenovirus is basically just the delivery tool, right? But if that delivery tool is stunted and it can't really replicate a little bit and expand your vaccine antigen, in this case the spike protein, it could potentially limit the immune response. So that's why they're using a chimp ad. So there's 4 of these recombinant in phase 3 trials. There's 3 in phase 3 that are subunit, a couple different versions here. So there's a Chinese company that's making just the spike protein, basically just take the spike, grow it in some cells in a petri dish, purify it, and they put it with some adjuvant, and that gets injected. So again, a subunit, just part of it. There's one from Novavax, a company I've never heard of, but called Novavax. They're using spike protein that is stuck onto nanoparticles. So using, again, these nanoparticles as sort of like a delivery vehicle to kind of get it into the cells. And then a really cool one, Medicago or something. They're actually have engineered a tobacco plant to make virus-like particles. So again, this is subunit. This is not a replicating virus or anything like that. It's just a virus-like particle. It looks like a virus to the immune system. It's like a shell but there's no genome inside. There's nothing there it's not an actual virus. But the tobacco leaves produce the proteins, and they kind of assemble into this virus-like particle, and it has the spike protein on it. And so again, that gets injected and can raise an immune response. So yeah, so there's 3 subunit in phase 3, and then there's 4 killed or inactivated. Again, historically, either weakening or killing it is the tried and true thing. And so there are 4 using that sort of older technology. Looks like there's 2 from a Chinese company. There's 1 that's being tested. Looks like the United Arab Emirates. There's also I think these all might be actually Chinese-based companies. A Sinovac. It's called CoronaVac. But yeah, there's 4 phase 3s around the world going on using killed or inactivated coronavirus. So you're literally taking SARS-CoV-2, but treating it in some way with, like I said, maybe heat, radiation, or a chemical to make it such that once it gets in the body, it cannot replicate anymore. So that kind of rounds it out. Josh, did you want anything while I went through that with some of this stuff, or have any questions? You can sort of be like so somebody might ask a question, sort of maybe like a non-biologist or non-scientist, right, from the general community.
Actually, I have a little more, I guess, in-depth type of question, because as you're going through that, it occurred to me, well, when they make a recombinant vaccine, is that the same technology then for the mRNA that is it really just the delivery mechanism now that makes that the big difference? So is that really, that hadn't occurred to me before, because we talk about the mRNA being such a new platform, and maybe it's not as new in my mind. I just never really thought about it as much.
Right. Right. So adenoviruses, which are, again, all 4 of the ones right now in phase 3 are all adenovirus based. So that's a DNA virus, right? It goes to the nucleus of a cell. Okay, so it gets in the cell, it's DNA based. So in the nucleus, just like I tried to show in that graphic before, in the nucleus, that's going to be transcribed by the host to make mRNA. That's going to go to the cytoplasm to be translated to make the spike protein. So again, you're just using the adenovirus as a way to get the gene, the DNA, into the cell to make the spike protein. Whereas the Moderna and the Pfizer, the mRNA based, you're just bypassing the delivery vector. They're just putting the mRNA into a particle and injecting that. So it's just sort of going right to the intermediate and not using any kind of delivery vector like a live virus. You're just going right to the nucleic acid, right to the intermediate, like right away.
And so, as you showed that graphic showing the spike protein on the outside part of the cell, that's essentially how the other vaccines would work as well, except for the protein-based vaccine, the subunit vaccines. You're actually just spewing that into the system and allowing the immune cells to pick it up and respond. Yes. Whereas the other ones are kind of basically hijacking our own cells to be the production crew. And that's why these mRNA vaccines are able to be produced in such mass quantities, because you only need such a small amount of mRNA to get into the person's system where they're making so much protein.
Right. And I think that's also why these came out faster, because I think the technology, it's a little bit more streamlined, a little bit more high throughput, a little bit more like plug and play, right? So like it takes a lot more work to take a gene out of adenovirus and put another gene in, right? It's just a little bit more labor intensive, a little bit more steps involved, versus just starting with just some plain DNA in a test tube. You transcribe that in a test tube to make mRNA, and you get rid of everything else, and you take the mRNA product, put it into this nanoparticle, and inject it, right? It's a little bit less intensive a process, I guess, and just a little bit more streamlined. So I'm wondering if these mRNA-based vaccines could potentially be sort of like the future go-to vaccine platform
That's what I was going to ask the question. Can we go after other diseases after this? After COVID is all said and done, can we go after other diseases?
Potentially, I think the answer is yes. Yeah.
Yeah, I think that's the most exciting part to this, is that if this works well, and you know, Lord knows we need it to, it'll be amazing to see what's on the horizon. All those different, you just mentioned how many companies, and once we can hopefully eliminate COVID, they'll have this platform and this mechanism to make vaccines for many different things.
Absolutely. And I'm sharing the screen again here, Josh. The New York Times website has a really nice tracker, you know, and you're mentioning this, Josh, just now. I mean, if you just look here, I mean, this is why basic research, basic science, this is why funding of science is so critical from just like a public health standpoint. I mean, if you look here, I mean, this came onto the scene sometime in late 2019, probably. We didn't really know about it until January, February, but it was circulating around the world probably last November, December. And here we are just about a year after. And look at this, Josh. Phase 1, 40, phase 2, 17, phase 3, 14. Those are the ones I just went through. And then 7 in what they call limited. So this is vaccines approved for early use. These are primarily the Russian and Chinese versions that didn't go through any kind of phase 3 for efficacy. They just passed the safety, and then they were sort of licensed in China and Russia.
Although in the UK, Adam, I just read that I think it was yesterday or the day before, this week anyway. The first patient in the UK got her first jab, they call it.
Yeah.
In the UK, I guess. So, yeah, it's all rolling so fast. And now imagine if all these, we could list maybe a separate disease. I've heard herpes simplex being mentioned as maybe the next obvious target.
Mm-hmm.
There's been talk about group A strep for ever since I've been in training. Cytomegalovirus. These are all ones that companies have had their sights set on, but now the infrastructure is there, and maybe we start to pick off all these other things. There could be a time in our near future where kids don't even have to worry about mono.
Right, exactly. And also too, it's the kids, and potentially, for example, like you mentioned cytomegalovirus, you know, if a pregnant woman gets that infection for the first time, it can cause severe birth defects. And so it's about kids, but also about protecting you know, kids in the womb too, and not just outside. I wanted to mention before we wrap up here, this was also on the New York Times website. I just saw this today. This is really cool. So it's kind of like a little tool, like an online tool. It says find your place in line, and I plugged it in. And so like it says, how old are you? I live in Montgomery County, Pennsylvania. I'm not any kind of essential worker. I don't consider myself a frontline teacher, not like in an elementary school or anything like that. And do you have COVID-related health risks? No. And you click submit, and based on my risk profile in this little tool, we believe that you're in line behind almost 269 million people in the US. And they have this graphic at the end here. And it says here, right, 800,000 healthcare workers, 86,000 nursing homes, right? So these are like the people that are theoretically in line to get the vaccine first. And if you keep scrolling down here, Josh, you just keep watching. Way at the bottom, there's me. So
I thought that was you.
Yeah. I apparently have scarlet fever.
Albright red. No, you're covered in Albright red.
Okay. So no, that was it's a pretty cool little. So they're taking these authors here are taking everything that's been released so far, I guess, on recommendations at the federal and state level of who might get it first, and kind of like a rank order list, and so they're trying to make an educated guess based on the information you put in.
That's interesting, Adam. We mentioned that I wanted to say, though, that might not be a bad thing to be a little further down the line in that line, because, we mentioned, how did you pronounce his name? Maurice Hilleman?
Yeah, Maurice Hilleman, yeah.
So we were in the last episode, there was discussion about how basic scientists have been left behind now, and so in the past things were named after scientists much more. But anyway, the father of modern vaccines, Maurice Hilleman, was quoted to say, "I never breathe a sigh of relief until the first 3 million doses are released," because you do worry that there's going to be something that wasn't picked up in the early studies, and like you mentioned, Adam, these things are followed very closely. And I mean, this now more than ever. I mean, the world's eyes are going to be on these vaccines. So, but it might be okay to be after the 3 million number, because then we're going to have even more information to ensure safety for all of us, and then we can jump in that line with even more confidence.
That's true. That's true. No, I mean, it's a good point, because if there's like a 1 in a million event, right? If you give it in some of these clinical trials, they're giving it to maybe 20,000 people. So you're likely, just because of statistics, you're not going to pick up a 1 in a million likely in a trial with 20,000 people. So yeah, but there's no indication at this point that these are not, I would say, very safe at this point.
Absolutely, and it's just that, as you know, and they try to make these studies are, like you said, Adam. They try to make these studies so they're very generalizable, so that the people that are going to get the vaccines are represented. But as you get more and more people and there, like you mentioned before, some people aren't. Some people shouldn't get the vaccines, but as time goes on, more and more people get the vaccines. Like obviously in these trials, patients are told not to get pregnant during these trials, but pregnancy happens, and so as that moves forward, we're gonna know about the safety in pregnant women, and that's not part of the study. It's not intended, but it's going to be information that we're able to get from it, and then we'll know how safe it is for pregnant women and for unborn children, for people with certain conditions, and then if you have that condition and you're worried about whether you should get the vaccine or not, now you're gonna have a better informed decision, and you'll know that, yes, I am going to be okay when I get this.
I'm really glad you brought that up. That's a really good point. Pregnant women, right? Because remember, my wife, when she got pregnant, she got a DTaP booster. So she had antibodies for tetanus and diphtheria toxin and for whooping cough. And the idea is she could pass it along through the placenta or the breast milk into the kid, right? And so I'm wondering, Josh, if these prove to be safe, especially, by the way, I would argue the mRNA ones, right? Because there's no virus at all. There's no organism. It's just this nucleic acid, and it's self-limiting, goes away. I almost wonder if an added benefit of that is you can generate antibodies, just like a DTaP booster for a pregnant woman. You can potentially have that woman be protected herself, but then also pass on antibodies to the fetus, so that when they're born, because remember, when a child's born, I mean, for the first maybe 2 months, 3 months, they don't really have much of their own immune system yet. They're relying on the antibodies and also the T cells they're getting from their mother through the placenta and from breastfeeding. So it's a cool thought that popped in my mind when you brought that up. So.
Well, and hopefully with the vaccine, this pandemic can come to an end quickly, and and we won't have to catch it. Not everyone will have to be vaccinated, and children will be protected because it'll be gone.
All I can say is amen to that. That's the hope. That's my New Year's wish, right? So anyway, I hope that going into the weeds just a little bit on the science here wasn't too intense, but I felt like it was important to kind of go through, like, what are the things being developed, a little bit about their background. So if anybody has any questions as they're listening to this or viewing this, please write in, and we'll try to address them on our next episode. But Josh, I think this will probably be our last episode of 2020, and 2020 cannot end soon enough. Yeah, so let's sort of reconnect on this maybe in mid-January, and we can maybe do another update on something, or just talk about whatever, right? That's the whole point of this. So but yeah, I'll give you the last word. The floor is yours. You want to what do you want to say to sign off?
No, I agree with you 100%. 2020 can't come to an end fast enough, I think, for most of us. And you hate to wish time away. You know, as you get older and older, you hate to wish your days away, waiting for the weekend. And you certainly hate to wish away a year. But if there was ever a year to wish away, I think we'd agree that 2020 would probably be high up on that list for all of us. So let's wish everybody an early Happy New Year, I guess, even though it's only the 9th of December.
Yeah. Happy holidays, everyone. Happy holidays, Josh. Happy New Year.